class of G Medical, and in today's class we are going to take a new chapter, very important and very simple chapter, mineral nutrition. So nutrition chapter or nutrition topic, you have heard, right? In your 10th standard you have heard about nutrition, the life processes. So there we have seen the various types of nutrition, how the nutrition is being taken or the nutrients are taken into the body, how nutrients are being utilized by the body, especially with respect to animals and human beings, right? But in this chapter, we are going to see nutrition with respect to plants, nutrients, how it is being utilized or how it is being metabolized inside the plant, right? So once again, welcome to the Mr. Vishnath, and we are going to start the chapter nutrition. So what exactly is mineral nutrient? Sorry. Mineral nutrient. So those are nothing but what? Some inorganic substances which is taken by the plants, which is going to act as what? So they're basically what? They are basically inorganic substances which is taken by whom? By living organisms. But since our chapter is about plants, we'll say it is taken by the plant, and organic substances, they are going to serve as what? Some raw materials. They are actually going to serve as raw materials for the synthesis of what? Some of the substances within the plant, or we can say going to be the part structure, etc. Various enzymes, it can be for constituents of the cell, anything it can be. So such inorganic substances taken by an organism which is going to synthesize or to make any component of the body, we call them as what? Mineral nutrient. And the taking in of this mineral nutrient, we call it as what? Mineral nutrient. Now, in this chapter, we actually discussing under various headings. So first, we will be studying about elements. Essential elements. So under essential elements, we see about number one is criteria for essentiality. Some things which is going to determine what quality of essential elements. Next, we will be studying about the types of essential elements. We are going to classify those essential elements. Essential elements. Next, we will study about the functions of these essential elements. Functions of essential elements. Then we'll bring out the deficiency of essential elements. Deficiency of essential elements, and we'll be studying all about toxicity, another thing, essential elements, toxicity of essential elements. Followed by, we'll be also studying about a technique known as hydroponics, and studying about nitrogen metabolism, exclusively about nitrogen metabolism. Nitrogen metabolism. Without wasting time, we will start with the essential elements. Essential elements. Essential elements are nothing but what? The elements that is required or with essential of the normal growth and development. Now, there are almost 105 elements discovered so far. 105 elements discovered so far. In that, more than 50 elements, sorry, more than 60 elements, they are found where? Down inside the plant. Now, we have seen that all 60 elements are of essential for the plants, right? There are some plants which will be actually taking in gold. Some elements will be taking the radioactive elements like strontium. Generally, they're going to take the radioactive elements. But we have to see whether this gold or any kind of radioactive element is of any function to the plant. Now, for that, we are actually going to discuss some criteria for essential elements. So, out of these 60 elements which is present in plant, we have to actually see how many elements are going to be an essential element and how many are going to be the non-essential element. So, not categorized into essential and non-essential element, there are certain criteria. So, that is what about the criteria for essentiality. So in your NCERT, they have given about 3 points. Those 3 points as such is very important because if NEET, the statement-based can which among the following is not a criteria for essentiality, or which amongst the following is a what criteria for essentiality. So you have to go through very thoroughly. So number one, criteria for essentiality. Number one is element must be absolutely necessary. The element must be absolutely necessary for the normal growth and development, or for the normal reproduction, growth, and development. Which means if these essential elements are absent, our growth, reproduction should be not right. So in absence of this, the plant should not, or plant will not. In absence of this, plant will not complete its life cycle. So clear? So in absence of that, no other activity should place inside a plant. Coming to the second criteria. The second criteria says that the requirement of a particular element should be specific. Specific means what? If element X is going to help in the growth, then no other one can help in what? Growth process. So the essentiality should be specific, which means in absence of an essential element, the plant should not what take it, or element can replace its position. It is like that much precious would be, right? If something is very precious, nothing can replace that particular thing. Same thing is applicable here. Essential element should be not replaceable. So example, if you're taking magnesium. So magnesium is actually the part of chlorophyll, right? You study this in photosynthesis in higher class chapter, structure of chlorophyll. If you're taking the structure of chlorophyll, it is having a head, it has a like structure. So magnesium is going to be in head region towards the center element. So if magnesium is not, then no one else can take the position of magnesium. Only magnesium can be there present in the absence of magnesium. This chlorophyll becomes what? Functional or becomes non-linear. So we call it as colorless chlorophyll. This was one example. Is it should actually directly involved in the metabolism. It must be directly involved in the metabolism. What do you mean by metabolism? Reaction, the sum total of all the reactions which is taking place inside a living organism. That is what we mean by metabolism. So clear? Essential elements, those elements, we call essential elements if they are going to satisfy the conditions which we have mentioned here. Now, based on this essentiality, or after taking this criteria, we found from there are almost 70 essential elements which are present in the plants. So plants are going to have how many? 70 essential elements, right? We classify these essential elements. Essential elements classification. Essential elements classification. So there are various criteria for this classification purpose. So the first criteria we take is amount present or the quantitative measurement. Is the amount present we are going to classify. So based on the amount present, there are two types of essential elements, namely macronutrients or macro elements and micronutrients. Macronutrients and micronutrients, these are the two types of essential elements based on the amount quantity which a plant is having. So we will take up in little bit of detail about this macro and micronutrients. Macronutrients and micronutrients. Right. Macronutrients and micronutrients. So macronutrients, as the name indicates, these are the nutrients that is required by the plants in larger amounts, whereas micronutrients are the one which is required in very small amount or trace. So they are required in large quantities or large, whereas micronutrients, if you are going to take, they are required in large quantities. Sorry, required in small quantities. Or we can say trace. That's why micronutrients are also known as trace elements, because quantity is very less. Now we will take up how much quantity it is required. It is required in more than 10 sorry, more than 10 millimole per kilogram of dry matter. What does it mean? If you are going to take a dry matter, 1 kilogram of dry matter, the amount will be what? It will be more than 10 millimole in that, the number particle size. Next one, just opposite of the less than 10 millimole per kilogram of matter. Now, out of 17, we said that after this criteria, we have got 17 essential elements. So based on quantity criteria, out of the 17 essential elements, 9 are coming under what? Macronutrients, and remaining 8 is the part of micronutrient. And we will write down these examples. That is very important. Just write down one by one. Hydrogen, carbon, oxygen, nitrogen, potassium, calcium, magnesium, phosphorus, sulfur. So this actually I have arranged in the descending order of their quantities, which means hydrogen is going to have highest quantity. The quantity decreases. Okay. Now we discuss the 8 micronutrients, which includes chlorine, iron, boron, manganese, zinc, copper, nickel, and molybdenum. Okay. Now, in this micronutrient, it was 16 essential elements. But as per modern studies, what happened? Nickel. They are actually newly introduced to this essential elements. And one thing you know here is iron. It was previously coming under what? It was previously coming under macronutrient. But later on, it was shifted from macro to the micro. So this arrangement, it is based on the decrease of particular names and examples of all the 17 essential elements is very important. Okay, mostly for NEET they can ask in medicine. So we come for the functions of. Now we will take up little more detail about the functions of micro and macro. Just the broad functions, particular functions we will take up. So the functions of both. So the macronutrient is actually going to be the part of the cell structure, we can say, okay, or plant structure. Part of plant structure. Whereas micro, it is not a part of the plant. Micronutrients are not going to be the part of structures or cell structure. As we said, it is a part of the plant structure. We can write one more point. They are part of the protoplasmic components. Of the protoplasmic components, and here we can say it is not a part of the protoplasmic components, not at all going to be, or very little, not we can say very little. Very little. Why? Because some of the ions can be dissolved in the protoplasmic component, so we can write very little. Okay. Now, they maintain the osmotic potential. What do you mean by osmotic potential? The osmotic potential means maintaining the balance of ions within the cell and outside to cell also. So there should be a total balance, or it should be in equilibrium. So that is maintained by macronutrient. Whereas micronutrient, it is not, but one exception is coming. We will take up when we come to the specific functions of each. Maintain osmotic potential. Now, I'm writing here. In 10th standard, you would have studied about photosynthesis, or again here we will study about photosynthesis next class. So, In photosynthesis, there is a process known as ETS, electron transport system. That's the system. Electron transport system. Electron will liberate out a molecule which will be taken by whom? By an electron acceptor. So from one electron acceptor to the another electron acceptor, it's like what? Moving. So it is going to form that pathway of electron acceptors, we call them as what? ETS. So the point what I want to say is, some of the micronutrients are part of the ETS. For example, copper is a part of the one of the component plastocyanin. Or if you're taking iron, iron is a part of what we say cytochrome. That is why cytochrome is ready. So the cytochrome as well as the plastocyanin, they are all electron acceptors, or they are the part of ETS. Okay, so the part of ETS. Example, we can write copper in plastocyanin. That's why it was in blue in color. That is why we call that blue gas. And we can also say cytochrome. Cytochrome, that is have iron. These are the two examples. Whereas macronutrients is not going to be the part of that. So very important point. So you can mark it as an important point. Macronutrients, as already I said, it is required by the plants in an amount. But if it is more than the required quantity, for example, if it is requiring 1 milligram mg, okay, but if going some point that is more than what is required, it has no problem with the plants. But in case of a micronutrient, already it is required only in very less amount. If it is required more than what is required, it can cause toxicity. There are two problems associated with this. We will discuss it together. So more quantity, which is usually what? Non-toxic. But with respect to micronutrients, if you are taking the more quantity, it shows us what? Toxic or toxicity, whereby there is toxic symptoms, toxicity. And if it is less quantity, then it shows what? Deficiency. So we can say the micronutrient should be in its actual amount. Whatever is required, only that much. Even a little increase is toxic for the plant. Now, the essential, this was what I teach for classification, which was purely based on what? Purely based on the quantity. Based on quantity, macro, macro, and small quantity required, and we have taken. Now, just briefly, because NCERT there is a mentioning about that. That's why just briefly only we have criteria based on function. Based on function. Number one, structural elements. Structural elements. Energy transducers. Energy transducers. Then the catalytic enzymes. Catalytic enzymes. And also we can say some elements that helps in osmotic potential maintaining. Osmotic potential. Now, based on functions, we are having what? Structural elements. Okay. Structural elements means which is going to be the part of the structure. Like for example, if you are going to see cellulose is going to be the part of the cell. So the cellulose is actually made up of hydrogen and oxygen. And there are some nucleic acids which is present inside the cell, inside the nucleus, which is made up of the nitrogen or nitrogen. So, which is going to be the part of the structure of the structural elements. Example includes carbon, hydrogen, oxygen, nitrogen, etc. Okay, which is going to be the part of structure. Like cellulose, or we can say forming the cell wall, or the DNA, RNA, and all, whereby nitrogen is also included. Coming to energy, they are the elements that is helping in transferring the energy from one form of energy to another form. I will explain with an example. Magnesium. Magnesium is a part of what? Chlorophyll. And what exactly is the function of chlorophyll in a plant? A plant's chlorophyll, it will actually capture light. That light energy ultimately is getting converted into what? Chemical energy. So that process we call as what? Transition. So magnesium, which is a part of the chlorophyll, in converting the light energy to the chemical energy. Another example we can say is the phosphorus, which is going to be part of ATP. Full form of ATP is what? Where is the phosphate group, phosphorus. Catalytic enzymes. Catalytic enzyme is, what do you mean by catalysis? Any compound which is actually speeding up the rate of reaction without involving in them. So there are some enzymes which will have some activators, which are nothing but our nutrients. Magnesium. So magnesium, if you see, they are actually helping for the activation of two enzymes, peptide and rubisco. The two enzymes you will see in photosynthesis chapter: pepco-phosphoenol-pyruvate carboxylase, and rubisco, you know the full form, that is ribulose bisphosphonates oxygenase. So this pepco and rubisco is having a metal, or we can say it is having an element activator, magnesium, right? And another one, molybdenum, that's going to activate the nitrogenase. So in this chapter itself, we will study about nitrogenase enzyme, because nitrogenase is acting for what? Nitrogen metabolism. It is involved in the nitrogen metabolism. And having in osmotic potential, we are taking the potassium ion. Because potassium ion, it is responsible for maintaining the osmotic potential in the guard cells. So the stomata is guard cells, right? It's protected, or we can say this is having a cell known as guard cell. And this guard cell is the one which is responsible for the opening and closing. When water enters into the guard cell, the stomata will be what? Opened. And when the water is leaving the guard cell, we will say the stomata will be closed. So that influx and efflux, that is inward movement of water, outward movement of water, is actually maintained by whom? By the guard cell with the help of potassium. So more details about this opening and closing of stomata we see in plants chapter. So just we can say potassium, opening and closing of stomata. Okay, opening and closing of stomata. So clear up to this much? Now, criteria is based on availability. So first one was based on quantity, and second one was based on what? Function. So we have to do with examples, and next one is based on availability. Number 3. Based on availability. So based on availability, we can say there are two types. The One is based on availability from and another one basically what we will say it will be available from where? It is available from the soil one, and another one is air or water. Okay. One it is obtained from soil and we call them as minerals. And second one which is obtained from water or air, which we call as the non-mineral elements. So these examples I'm leaving it to you. You can go find the example. And next one is what point I want to tell you. If you're taking nitrogen, it can be obtained both from the soil as well as from the air. So it can be both mineral and non-mineral. So this is why we always say nitrogen is partially mineral nutrient. They are partially mineral nutrient. Now next one, based on mobility. Next classification. So based on mobility, the ability of an element to move from one position to another. So based on the ability of an element to move, there are two types of elements. One will be the one which is having the ability to move, because mobile element, and another one which is not having the ability, we call them as what? Non-mobile elements. So two. Mobile elements, that is, it can move from where? It can move from an olive to the tip of a growing young leaf. Okay. So nitrogen, magnesium, and all we can say as an example. Next one is non-mobile elements. So the non-mobile elements, we can say they are not having the ability for moving. Example, very important non-mobile element we are taking is calcium. Why? Because calcium is actually a part of a structural component of the cell. In cell chapter, you would have studied about the middle lamella. So calcium is going to be a very important component in that. So hence we can say the calcium is not able to move from one position to another. Hence under which category? Non-mobile. Now, when saying about essential elements, I would like to introduce a term to you: critical elements. Critical elements are basically very lessly found in a particular way. Okay, they are very less found in an agricultural soil. Which one? Critical elements. There are two reasons for that. What are the two reasons for this concentration of the critical elements in the soil? Number one, we can say due to the runoff with water, which we call as leaching. By the process of leaching, they can what? Washing away of this particular elements from soil. And another one is by the absorption by the plants. So plants which is growing can take this element. Absorption by the plants. So I am citing few examples of critical elements: nitrogen, phosphorus, and potassium, some of the very important critical elements. So their deficiency, okay, causes some symptoms in the so which we call as the primary deficiency. So, and any fertilizer which is having these elements are considered as what? Complete fertilizers. So this is a term, critical element. Critical elements. Now we will cover. Hope it was clear up to this much. So we were discussing about the essential elements. So we have taken the criteria for essentiality. Based on the criteria of essentiality, we got 17 elements. And that 17 elements can be further classified based on certain criteria. Criteria number 1, what was that? It is based on the quantity, how much quantity a plant is having. We are having macronutrient and micronutrient. Second one, based on what? Based on the functions they perform, we are having the structural element and so 4 categories. It is based on whether it is or not. We are mobile and immobile elements, and also we are having what? The elements that are actually, what was that? Based on the availability from the soil or not obtained from the soil. Now we will take up the macro and micronutrients in detail. So we will take up macro. So basically, very easy to study the functions of these macronutrients is nothing but making a tabulation. So we have almost 9. In that we will be taking detail because those are the one which is sources from the soil. So the absorption forms we have to study and the functions of them. And deficiency will be how mentioned in the NCERT. So the one by one we will take up. Erasing in between because of the space constraint. One we take up nitrogen. So in nitrogen, we will study how it is absorbed. And functions. Okay, nitrogen, it is absorbed in form of NO3, nitrate. So sometimes as nitrite and ammonia also, but mostly the plant will be preferring in the NO3 form, nitrate form. We know that nitrogen is an important component of proteins. The amino acids are having nitrogen. Amine acids, amino group, NH3 group will be nitrogen. If you're taking previously, I have said the particle RNA and all, whereby nitrogen is having the nitrogen base is coming. So we can say part of nucleic acids, vitamins, and few hormones. Then next to phosphorus. Phosphorus. So the phosphorus, it is attached as phosphate ions in the H2PO4 minus or HPO4 2 minus. And the phosphorus, it is a part of what? The cell membrane. In cell chapter you will study, right? The nickel, zinc, we said it is what? It is layers. Right, 5 layers where phosphate is also be a part of that. So we can say it is a part of cell membranes, part of the cell membrane. Again, nucleic acids, DNA and RNA is having what? The nucleic acid taken in the form of phosphoric acid, right? Nucleic acid. And for all phosphorylation reaction, ATP formation reactions under comes under phosphorylation. phosphorylation reactions will have that. Energy molecules like ATP will have what? Phosphorus. Next, calcium. Calcium. So the calcium is taken in the form of calcium ions. It is taken in the form of calcium ions, Ca2 plus. It is going to form part. We already said that it is immobile because of this reason. Part of the middle lamella. Mitotic spindle fibers. In cell division chapter, you will study about the spindle fibers. Mitotic and also it activates certain enzymes. It activates certain enzymes. Okay. Calcium. Next one, magnesium. Magnesium, again it is taken in the form of magnesium ion Mg2 plus. Already we said it is a part of the. At the same time, we also said that it is involved in the activation of certain enzymes. Activation of certain enzymes. Okay. And synthesis of DNA will have requirement. Synthesis of DNA. Next one is sulfur. Sulfur again take off sulfate form. Okay. Amino acids, sulfur containing amino acids like cysteine, methionine, etc. Methionine contains this, and it is the part of coenzyme vitamins. Few vitamins part it is will be okay. Biotin, thiamine, etc. Thiamine, etc. Okay. Now I am erasing nitrogen, and here we will write all of it. So again, it is absorbed as what? K prion. Function. We already said that it is helping in maintaining the osmotic potential. It maintains the pressure. It's in the opening and closing. And we said that how based on the turgor pressure, the opening and closing of stomata will take place. Also involved in the protein synthesis as well. So now we move to micronutrients. So these are the role of macronutrients in the plants. So nitrogen, phosphorus, calcium, magnesium, sulfur, along with and functions. This is very important. Matrix match questions can be asked. They will be listing out the names of the elements Absorbed form, they may not ask because as we can about function. So which one correctly matches, well, you have to find out the combination of. So like that questions can be asked in NEET. So moving on to the next one is micronutrient. Micronutrient in the form, absorbed form, and get of functions. The absorbed form and the functions we will take. So first one we are going to take iron. Iron. So iron, it is absorbed as ferric ion. The ferric form, Fe3 plus ferric in the ferric form. Now it is that it is a part of ETS, right? Cytochrome is having iron in them, so it is a part of ETS, right? Electron transport system. We already said that how electrons are accepted from one to another. In that one electron acceptor is cytochrome. Cytochrome is having iron inside them. Okay. It's part in chlorophyll synthesis. For making of chlorophyll. So you should understand one thing. Chlorophyll does not have magnesium and other elements in that, but iron is required for the synthesis of chlorophyll. That is very much important point. You don't get confused. Okay, iron is not a part of the chlorophyll, but a part in synthesis of chlorophyll. It is a part of. I am writing chlorophyll synthesis. It is a part of the chlorophyll synthesis. And it also activates certain enzymes, metal activators, certain enzymes. The detailed part and not we need not to go. Okay. Strictly, you read up to the NCERT level. That is only required. Okay. Moving on to the next one is manganese. Manganese. Manganese is what? It is taken in the form of Mn2+. And in photosynthesis chapter, you will study photosynthesis requires water, right? The equation is carbon dioxide plus water. So that water in the photosynthesis is actually a electron donor. It requires a electron. So in order to donate electron in this, has to be there should be splitting of water. So the splitting of water takes place in presence of light. So the water molecule, it gets in presence of light, right? So in presence of light, this water is getting splitted and oxygen is right, along with some protons and electrons released. Now this electron, it is actually intake what is used in the process of photosynthesis. We will come in the next class, photosynthesis in higher plants. So the splitting of this particular water into oxygen and electrons and protons, an element is required known as what? Manganese. The process of splitting. Splitting another word, lysis. So here the lysis of water is taking place in presence of light. For light, we also use the term photo. Photosynthesis is something in presence of light. So yeah, light is used for lysis of water. So we can call this process as what? Photolysis of water. So in the photolysis of water is in presence of light into the respective protons, electrons, and oxygen is also released. Now that requires what? Manganese. So that can be the first option. Activate, or it is involved in photolysis. One second. It's a part of photolysis. Complex called as OEC and all. Right now, don't get with that. Photolysis of water. Another one, we can do the same function, activate certain enzymes. Next one is zinc. Zinc and zinc. Very simple one function we have to notice. It activates enzymes that are involved in this. Involved in synthesis of oxygen. Next one is copper. Taken in Cu2 plus form. Again, it can say all metabolism. Copper is involved in all metabolism. And also we have previously taken what? Component in the ETS. Cyanine, I said that, right? So in the plaster cyanine, copper is going to be an important part. So we can write that also. Part of ETS. Copper. Okay. Next one is boron. I'm using this. Boron. Boron, it is absorbed in the form of BO3 or B4O7 2 minus. Okay. And boron, it is very important element in the absorption or intake of calcium. Okay. Utilization of calcium. Also, it is called in cell elongation, cell differentiation, etc. Cell elongation and cell differentiation. Even transport carbohydrate called carbohydrate translocation. Transport of carbohydrate or the carbohydrate translation. Fine. Next one. Sorry. Boron, right? Okay. Next one. Molybdenum. Absorbed in the form of, I'll erase that. So molybdenum. Molybdenum is actually absorbed in the form of MoO2 2 minus ion, and already we said that it is helping in what functions? It is helping in activation of enzyme. Which enzyme? Nitrogenase, which is used in nitrogen metabolism. Okay, and this nitrogenase is used in the nitrogen metabolism. Okay. Now we move the topic. So we were discussing about the criteria. What was the first criteria for the classification of essential elements based on the quantity? So macro and micro. So also take 7 essential elements classified into the macro and micro, and their essentiality role we have taken. Absorbed form as well as the function. This is very, very important. Okay. So in NCERT it is listed as just point by point, line by line. Okay. Now next topic, deficiency of essential elements. So these were the functions of essential elements. Now next is the deficiency of essential elements. Deficiency of essential elements. So whenever, even if in some cases due to the deficiency of vitamins, we also develop symptoms, right? So we will have like some of them, like for the deficiency can cause to bleeding, mouth bleeding happen, or various things happen in case of animals. Similarly, this deficiency of some of the nutrients in the plant, it can also cause some symptoms which can be externally. Morphologically, it is as well, deficiency. So the sum of the essential elements, we already said that essential elements cannot be replaced, right? The function of calcium can only by magnesium cannot replace calcium. Needs to do its function. So everyone has to do its own functions, and no one should bother about what others are doing. So the same thing is applicable. So when it's a deficiency, what should we understand? There are which cause a particular deficiency. Just like if we have some symptoms, we will say that okay, we got that disease. Similarly, if there is deficiency of element, there are symptoms which is already found out. Found we take in the upcoming topics. So deficiency. So the deficiency of essential elements. So before going on to the topic, I'm going to say about a very important term in this, which is critical concentration. Very important term which is known as critical concentration. So critical concentration can be said as a concentration of essential elements below which plants will develop deficiency symptoms. Okay, so write down the definition. Concentration of essential elements below which the plant or the plant growth is inhibited. That will be better word. The plant growth is inhibited. That is what we mean by the what critical concentration. Now, if concentration is the concentration of our essential element, if it is below the critical concentration, definitely what the plants will develop a certain deficiency. Less critical concentration. What will happen? We can say that there will be a deficiency. Deficiency symptoms will be developed by the plants. So the deficiency is not purely based on the critical concentration. Okay, deficiency symptom also can be on the basis of mobility of elements. Can also be based on mobility of the elements. So previously we have taken a type of essential elements based on mobility, right? We are having some elements which is capable of moving, known as mobile elements, and those which is not having that ability, immobile elements. Take an example here. I'll explain it with a diagram. So taking a potted plant. So the leaves I have drawn, and obviously from the size or from the position, it is very much clear that this is going to be the outer leaf, and this is going to be the inner leaf. Inner leaf. So the outer leaves are having certain elements using the red color dots, which we are calling them as the mobile elements. It is having mobile elements. So as indicated, also we said that mobile elements are having the ability to what? Move from one place to another. So the older leaf is having some mobile elements. So what will happen? Due of time, this mobile elements, it tend to move from the older leaves to the growing of the younger leaves. You have written that already. So as a result, what will happen? The deficiency of mobile elements can first happen to whom? To the older leaves. So we're going to take up with respect to mobile elements. Okay. So in case of mobile elements, the deficiency symptom occurs first in outer leaves. This is a very important point. Okay, observes in outer leaf. Why? Because more nutrients are required for synthesis of younger leaves, right? Already what has happened. So now whenever younger leaf is requiring, the mobile elements will move after. Okay, older leaf is done. So now I have to go to, so mobile elements will move to the younger leaf. Now in case of a non-mobile element, this is opposite. Just like what we say, deficiency symptom first occur in younger leaf. Because younger leaves, they are going to be a part of structure. It's a forming stage. It has not formed yet. So deficiency symptoms first is observed in the younger leaf, then older. So this is two important things. They can ask examples. So they can ask calcium deficiency is first observed where? So calcium is a non-mobile element. So definitely we can say fish will stop in the younger leaf and then older leaves. Now we will take up the some of the deficiency symptoms. The deficiency symptoms. Deficiency symptoms. So we will take up one by one. Number one is chlorosis. Chlorosis. So chlorosis is actually yellowing of the leaf. Due to destruction of chlorophyll, one chance is there. due to the destruction of the chlorophyll can be of the leaf, or due to the non-synthesis of chlorophyll also. So or due to the non-synthesis of chlorophyll, the leaves can become yellow in color. And yellow leaves, you know that they are not suitable for doing the process of photosynthesis and ultimately chlorosis. Necrosis. Necrosis. So, as we already said that yellowing of the leaf can cause damage to the leaf, thereby there won't be any photosynthesis happening, and ultimately due to the yellowing of the leaf, what can happen? The death of the leaf can happen. So the process of a death of leaf to process process we call as what? Necrosis. So necrosis is going to represent the death of a tissue. Then disproportionate growth, or we can say not growing properly. We use the term stunted growth. Now, the young leaves as well as the bud, it can fall off before what? Its maturation. Actually, if as well as the bud, it should completely and should do its functions. But lack of that full complete growth results in the early forming of the leaves as the buds. So we call it as premature falling of leaves and buds. Leaves and buds. And next one is delayed flowering. So these are the some of the very important deficiency symptoms. Now we write the names of some of the elements responsible for this. Nitrogen, potassium, sulfur, manganese, magnesium, zinc, etc. Okay. In this, nitrogen, potassium, sulfur, and manganese, they actually causes what we can say it is going to affect the cell division. Initial cell division. Cell, which are nitrogen, potassium, sulfur, and molybdenum. They are actually responsible for what? Initial division. And the next three of them, that is nitrogen, sulfur, and molybdenum, they causes what? Delayed flowering. Okay. Necrosis, calcium, magnesium, potassium, and copper. These are the elements caused. Clear? So the some of the deficiency symptoms include what? Losses. It's not the yellowing of the leaf, either due to destruction or not in the chlorophyll, and the elements involved in the chlorosis process is listed here. Necrosis, it is death of the tissue, and magnesium, potassium, and copper. And it is calcium and all can be an example of that. Premature falling of the leaves and the buds and delayed flowering. Okay, these are the common deficiency symptoms. Next one is micronutrients. toxicity of micronutrients. So in the beginning, when we discussed about the types of nutrients based on amount, we said that a slight increase in the concentration of micronutrients is toxic. The system, its deficiency also causes limited intake also causes deficiency, right? So the micronutrients, if they are having a moderate low concentration, it causes what? Symptoms. If it is moderate in higher concentration, it causes what? Toxicity. Toxicity. Now, plants generally, if we take, it is very difficult to find out the toxicity in plants. It's very difficult to find the toxicity. The major reason for this difficulty is the toxicity of one micronutrient. It can even cause deficiency of other nutrient. Are you getting this? For example, if one nutrient is toxic due to their over presence, it can cause deficiency of others. So it is not actually clear what is the deficiency symptom or if it is a toxicity symptom. I will explain with one example. A micronutrient. So let us take example manganese. Manganese excess element causes some toxicity. Now, due to the more presence of manganese, the concentration of few other elements, namely iron, magnesium, and calcium. So the concentration of these three elements, what will happen? It is reduced. So the toxicity of a micronutrient can cause the deficiency of some other nutrients as well. If you're taking example manganese, so manganese toxicity, manganese concentration is more inside the more concentration of manganese can lead to the lower concentration of what? These three elements. So what is the reason for that? Probable reason for this is that manganese will actually compete with iron and magnesium while intake. Okay. It competes with iron and magnesium at the time of intake. So obviously more manganese intake can result in reduced intake of iron and magnesium. This is one reason due to the toxicity of manganese, the deficiency of iron and magnesium. At the same time, manganese toxicity can actually block the translocation of calcium to where it is required. It blocks movement of calcium. So I said in plants, it's generally can find the toxicity because the toxicity of one element can lead to the deficiency of other elements. If you are taking manganese toxicity can lead to the reduced concentration of iron, magnesium, and calcium. Two reasons are there. Reason number one, manganese actually competes with iron and magnesium. So if manganese is more, obviously the iron and magnesium concentration or magnesium intake by the plants will be reduced. At the same time, manganese will actually block the movement of calcium. In general, taking its amount will be reduced. So clear? So this is all about what? The essential elements. So in essential elements, first we have taken what? The criteria for essentiality. Then we take the types of essential elements. Then we have seen the functions of essential elements, deficiency symptoms of essential elements, and toxicity. Now we will move on to the second very important topic of this chapter, that is hydroponics. Hydroponics. It was given by the scientist Julius von in the year 1860. In the year 1860. So, if you're taking into history, Aristotle, in almost in the 13th century, around that time, he said that, which you may feel like something rubbish, plants are made of soils completely. Instead of the complete soil. So it is the soil which later decides whether it has to form a branch, flowers, or fruits accordingly. But in another perspective, he said is actually correct, right? Plants is made of whatever is present in the soil. And later on, it by different studies, especially after hydroponics, it's not the soil which is very important. It is what is in the soil which is important for the growth of the plant. So by hydroponics, Julius von Sack demonstrated that plants can be grown in the complete of soil. What we have to do, the nutrients for the plants is given in the form of a solution. So with the help of the solution, the plant is to be in the complete absence of the soil. It is not the soil, what is in the soil that is going to determine the growth and development of the plant. So plants can be grown in a defined nutrient. In hydroponics, plants can be grown in defined nutrients in complete absence of soil. And the solution are actually given in the form of a solution, and that solution we call as balanced solution or normal solution. Balanced or solution. Okay, so we will see how a hydroponics will look like. It was once a previous question. They have given the diagram and asked to label. So let us A and B, and we have to find out what is A and B. So the hydroponics is essentially consists of what? A container So let's say this is a container. The container, it is actually insulated with cork or something, with cork and all. That is just to place the equipment as well as the plant. Just insulated. Now to this container or we are introducing a funnel. Now it is through this nutrients we are pouring into this. So we have now nutrient solution. Now to this we are introducing the plant. Correctly, we are introducing with the developed roots and all. Introduced to this. Now, along with that, another tube we are introducing to this. And we call it as aeration. This is the aeration tube. So the aeration tube is one which actually maintaining or providing air to nutrient solution, and the nutrient solution it will be changed in every 2 to 3 days time as well. Okay, and all the balance are given. Now, this is the significance of hydroponics. Other uses. Using hydroponics, deficiency symptoms can be observed. Deficiency symptoms can be observed. How? For example, that solution we are giving externally. So we know that 17 essential elements are actually are required for the plants. So what we will give? We will give 16 essential nutrients, and people are not giving nitrogen. So we know that who have given that nutrient solution knows that nitrogen is not given. So when nitrogen is not given, we will get one plant with complete nutrient and without nitrogen, remaining all 16 we are giving. And we will compare both the plants. One plant with all the 17 essential elements is healthy, and another one is having different types of like necrosis and chlorosis happens. So what we can say, in the absence of nitrogen, chlorosis is there. Similarly, we give nitrogen, we won't give what potassium, and the plant having standard growth and all such complications will observe. So this way, by this method, we can study what deficiency symptoms of various. So we list some of deficiency symptoms that will be probably calculated by this particular technique. Now, this we also can understand which is essential and non-essential element. Which is essential and non-essential element. By this technique, we can know that as well. How? The same way. We will provide and we will see beneficial symptoms there in that absence. If it is not there, definitely that it is not much required for the plant, so it is a non-essential element. Apart from that, it also can be used commercially for the production of various compounds. Sorry, various crops. So we will take up that commercial. So the hydroponic technique, which is used commercially, okay, we call it as thin layer hydroponics. Thin layer hydroponics. So in NCERT there is a diagram about the thin layer hydroponics. So I'll just draw the diagrammatic representation of it. a tube, and the tube is in slanting position. Okay, and in between the tubes there are some cavities, and this cavity is inside like previous way for holding the plants. Proper diagram is there in NCERT. So this is just for that. This is for just understanding purpose I'm drawing. So stand. Now this one end of the tube, it is opening into a into a tray, it is opening. Now in the tray, so we are actually mod. Okay, so this tray is actually holding what? The nutrient reservoir. The nutrient reservoir will be there inside this. Now what will happen? We are also introducing plants. Okay. So what with help of the motor, the reservoir is pumped to the another end of the tube. So with the help of the motor, it is actually pumped to the another end. Now what will happen? It is already in an inclined position or slanting position. Now the reservoir will reach the plant, whereby continuous absorption will take place, and whatever is left out, it will reach back to where? To it is continuous a cycle. It is happening, which means the reservoir is not wasted unnecessarily. Okay, so this is the thin layer one. Okay, now this one is we are actually doing. We are actually used for the where this type of thin layer hydroponics is usually used in soil is fertile, sorry, infertile, lacking the nutrients, and it's used in the production, commercial production of tomatoes, cucumbers, lettuce, seedless cucumber, etc. So this was once a question also. Okay, this hydroponics is used for that. So this was one option. Okay, it's very important. Now, in your NCERT, two more topics are mentioned. That is translation of nutrients and absorption as well. But right now, I am not taking that. Why? Because in transport in plants, we are going to study the same thing in detail. When study that, we will go into those detailing part. Right now, that I'm okay, because in transport in plants we will be taking that absorption and all different methods of absorption, active, passive, and all we will take up that. Now we'll move to the last topic of this chapter, that is nitrogen metabolism. Nitrogen metabolism. Now nitrogen found in the atmosphere, which is going to be around like 78, 79% it will be present. But the nitrogen is present, it cannot be taken by the plants directly. Before that, what is the function inside a plant's body? We already said the nitrogen is a part of what? Proteins. For amino acids, nitrogen is there. So this amino acids are joining together and form a protein. For the synthesis of amino acid, for the protein requires nitrogen. Nitrogen is an important constant of the DNA and RNA or the plasmid. So without nitrogen, it is not possible for an organism to survive. On the other hand, we can say nitrogen is very much present in the atmosphere, almost 87% present, but unfortunately, that nitrogen cannot be taken by the plants directly. So in order for that, they should be actually converted into usable form. And that usable form, the plants will absorb and an animal will eat the plant and it is coming to the animal. So that way it is actually happening. So the process of conversion of the atmospheric into usable form, we call that process as what? Nitrogen fixation. So nitrogen fixation is going to be important in the nitrogen cycle. So in nitrogen metabolism, first we will take up the nitrogen cycle. Again, a diagram is given in NCERT. So there also that same diagram it has been asked, and gaps empty, and they will be writing A, B, C and all, and you have to find out which is A, B and C. So the diagram, NCERT diagram, which is about is also very, very important. Okay. So first one, nitrogen cycle. So as I already said, we are having what? Having atmospheric nitrogen. Atmospheric nitrogen. Now, this atmospheric nitrogen has to be converted into what? As I said, usable form. Because atmospheric nitrogen cannot be taken by plants directly. So it has to be converted into a usable form. And that process of conversion we call as what? Nitrization. We will leave out the name, or we will take up the usable form. So usable form can be ammonia, but the ammonia as such is not taken by the plants. So the ammonia will be converted into nitrite. NO2 is nitrite, and from nitrite it is going transferred to where? Converted into nitrite. And this nitrite will be converted. So before that, the atmospheric nitrogen, it is converted into the usable form by three methods. So one is biological nitrogen fixation. Biological nitrogen fixation. Second one is industrial nitrogen fixation. Industrial nitrogen fixation. Third one is the electrical or the physical nitrogen fixation. Or the physical nitrogen. Now, as I said, the usable form, mainly the nitrite, it will be absorbed by plants. It will be absorbed by whom? By the plants. And these plants, they are eaten by ants. So in that way, now the nitrogen is inside whom now? Inside the animals. Now what will happen? The plant as well as the animals, over time, what happen? They will die, which means they can decay. Dead and decay. Okay. Over a time period, it will be dead and decayed. Now it has, along with that, what will happen? The animal also excrete nitrogenous waste. Like urea and all. Urea and all other nitrogenous forms animals will. Now, this dead and decayed component will have nitrogen as well as the excreta also will have nitrogen. So this compound, they will be converted back to ammonia. It will converted back to ammonia, and that process version of nitrogenous compound back to ammonia as ammonia, which is done by the ammonifying bacteria. Now it's a cycle. So if continuously atmospheric nitrogen for purpose, there can be deficiency of atmospheric nitrogen and cycle won't be complete, right? We say water, the water in the body is evaporated, condensed, and it is coming into recharging again. Here in this diagram right now, it is not getting back. So what will happen? With the help of bacteria, some of this ammonia after ammonia will go back into the atmosphere, so which we call as the go back to the atmosphere. So this is how the nitrogen cycle takes place. In this chapter, in the nitrogen metabolism, we'll be detailing about biological nitrogen fixation. But before that, I'll just say about electric nitrogen fixation or the physical nitrogen fixation. Here, the nitrogen molecules can get splitted up used by the lightning or any other physical forces, and nitrogen, which is splitted, it with the water. It joins with the water, and this water will actually reaches the earth acid, or what we can say nitric acid or nitrous acid. And as a acid reaches where the nitric acid and the nitrous acid will later back dissociate into nitrate or nitrite. And nitrate, we know it's a usable form, will be taken by the plants. Now, the plant, as I said, it will be absorbing what? The nitrate. Inside the plants, nitrate will become again ammonia, and from ammonia, amino acid formed. So we will study about that. Nitrogen will end up. So that is what happens there. Okay. Now, in this chapter, we will deal about, as I already informed, biological nitrogen fixation. So we'll take a biological nitrogen fixation now. So, nitrogen fixation. So something regarding biology. What exactly is that? Using living organisms, some kind of nitrogen is fixed. Mostly it includes living organisms. Living organisms. And living organisms which is used for this biological nitrogen It can be broadly divided into two: symbiotic and okay. So it can be two types, as I said: symbiotic and non-symbiotic. We can write asymbiotic. We can also call the term they are free living, which means it is not associated with any organisms. It is free. And we can say next one is what? Symbiotic. Symbiotic means mostly it will be found in association with the roots of the plants. Roots of the plants. Now we will take asymbiotic. The asymbiotic example can be remembered with the code word A, B, C. A stands for azotobacter. B stands for Bajeringia. And C stands for Clostridium. Clostridium, okay? Now, in this, the first two, that is Azotobacter and Bajeringia, they are aerobic. And Clostridium, they are anaerobic. Anaerobic. Moving to the symbiotic one, we can say example of a bacteria, Rhizobium. And as we said, the symbiotic type of this living organism will be found associated with the roots of the plants. Here we can say it is found in the roots of leguminous plants. Leguminous plants. And there are non-leguminous plants also. So the non-leguminous plant is having an organism attached in the roots, that is Frankia. The roots of non-leguminous. Okay. Now, another group of organisms in biological classification has under kingdom Monera, that is blue-green algae. Blue-green algae. Right. Now, blue-green algae, they can be both symbiotic or free-living. Now, it can be found living, so alone, or we can say non-symptomatic, asymbiotic, free, as well as it can be found inside some of organisms we already discussed there, the roots and all also it is seen. Okay, that is symbiotic relationship of that bacteria. Now, we will go into the detailed part of the biological classification, that is nodule formation. Take example of the Rhizobium leguminous plant and all. The entire diagram drawing, it is not practical, so you have to just see the NCERT with the diagrams given with the stage and all. That comes like a seat to identify what is the correct seat. So different steps of this particular nodule formation. Okay, so let's say this is a leguminous plant. So when this plant is in the growth stage, it will release certain chemicals into the soil known as flavonoids. Plants. Now, they were in the soil. So this Rhizobium will detect some flavonoids, due to which it will move the to the region where it is secreted, that is especially towards where? Towards the what? The roots. So it is, or it causing the movement, chemotatic movement. You'll study movement. So the rhizobium, it is moving to the region where the flavonoids are released, and flavonoids are released by the leguminous at the time of maturity or during the growth process. Now what will happen? This microbe, it will colonize near the root. The root are having tiny unicellular microbe. In anatomy chapter, you have studied the growth, unicellular outgrowth, which is a part of the epidermal tissue, epidermal outgrowth. So this epidermal outgrowth, we call them as what? The root hairs, which is unicellular in nature. So what will happen due to the colonization of the rhizobium near the root? Root hairs will bend. The tips will get bent. That is actually making the entry of the rhizobium more easy. Now, what will happen later on? The rhizobium enter root through root hairs, either by two ways: node entry through the to the tip, or through the side of the crack entry. Out of these two, mostly the rhizobium will be entering through the nodal region. That is the tip of the root hairs. Now what will happen? This particular rhizobium, it will form a connection reaching the cortex of the root. You know that cortex region of the root, right? In a dichotomous, again, you studied that. So what will happen? There will be a structure which is extending up to the cortex, and the rhizobium finally reaches the cortex. And this thread-like structure we call as now what will happen? At last, the rhizobium is now where? In the cortex. And the cortex, the cortex of root is generally made up of parenchymatous cells. We have studied that. So now the cortex is having parenchymatous cells. The rhizobium is reaching this cortex. So what will happen? The parenchymatous cells of the cortex will be now colonized by the rhizobium, and they convert the parenchymatous cells into specialized nitrogen-fixing cells. So previously it was just a normal parenchyma cells, but now rhizobium is reaching the parenchyma of the cortex region. So now that parenchyma cells nitrogen fixing cells. Okay. So the rhizobium, it enters the root through node entry and crack entry, and finally reaches the cortex. And cortex was having normal parenchyma cells. Now what will happen? The parenchyma cells is converted into specialized nitrogen fixing cells. And the specialized nitrogen fixing cells, we call them as what? Nodule. And a mature nodule, if you see, it is pinkish red in color due to the presence of common as leg hemoglobin. Leg hemoglobin. What is leg hemoglobin? So, hemoglobin, just about hemoglobin. So, the hemoglobin, actually what it does, it carries the oxygen. In hemoglobin, they are actually oxygen scavengers. They will take away the oxygen. And why that oxygen has to be scavenged there? Listen, for the process of nitrogen fixation, It's a requirement of an enzyme known as nitrogenase. And this nitrogenase is seen in we have taken the example. So this nitrogenase enzyme, it will work only in will be inactive in stage. It work in anaerobic condition. Okay. Now, for the if nitrogenase enzyme is working only, nitrogen fixation can take place, which means the cells that is going to fix, especially that oxygen is not there. So it is normal parenchyma cells, so there can be chance where nodule were present. So now, in order to avoid that oxygen, take away the oxygen from the cancer, we are having what? Like hemoglobin literally will scavenge that oxygen so that that place is suitable for the nitrogenase enzyme's activity, which leads to extinction. So that's all about the process whereby the rhizobium is colonizing. So the root, it will release the flavonoids, due to which rhizobium will colonize near the hair, and that hairs will have a bended and it enters by two ways, node entry or crack entry, and forms an infectious thread leading to where? To the cortex, and in cortex the specialized cells form as stool, which is pink in color. Hemoglobin. Clear? No. We will see the nitrogenase enzyme activity. So in the chemicals chapter, the enzymes are having various shapes, right? It is having an active site, right, for where the substrates are attaching and all. So I'm drawing a shape of a nitrogenase enzyme having the active site. To this active site, what will come and attach? The nitrogen will come and attach. So nitrogen, know that it is bound to each other by 3 bonds. Nitrogen. Now, as soon as this nitrogen is attached to the nitrogenase enzyme, what will happen? There will be addition of 2 H plus and 2 electrons. Okay. As a result, one bond which is actually called 2 nitrogen will become, and that will be occupied by whom? By the hydrogen. So after that, now the nitrogenase enzyme complex will be like this. So it will happen. Our again nitrogen. So previously, how many bonds were? 3 bonds. Now in that 3 bonds, that will be taken by whom now? Hydrogen. 2 electrons and 2 protons. Again, an addition of 2 H will come, 2 H plus and electrons. So again, the nitrogenase activity will repeat. So previously 2 bonds and 2 hydrogen, 1 hydrogen was there. Now what will happen? The second bond of the nitrogen is broken now. So finally, how many are there? 1. So it's nitrogen. This is nitrogen. How many will be there now? Previously, one hydrogen was there. Now, additionally, another hydrogen is also added. Again, it will happen. Again, 2 hydrogen and electrons are added. 2 H plus and electrons are added. So again, the nitrogenase activity continue. Now, the bond of the nitrogen is broken. And as a result, finally, how many hydrogen? 3 hydrogens. So, from a single molecule of nitrogen, we are finally getting what? 2 molecule of ammonia. Along with 2 that is, there is production of hydrogen. Hydrogen is a byproduct here, and that hydrogen synthesis actually. So the hydrogen is byproduct, and that byproduct is formed using 2 electrons and 2. So we'll find out the total equation of this nitrogen using the nitrogenase enzyme. So there was one molecule of nitrogen. One molecule of nitrogen have taken how many? First constant 2H plus, third one again 2, 6, and for byproduct how many? 2H plus. So total of how many H plus? 8H plus. And along with that again 10 electrons, 8 electrons. And one more thing you have to keep in mind, for 1 electron transport, okay, for 1 electron transport there is requirement of 2 ATP. So here actually how many electrons are there? 8 electrons. So 8 electrons is how many ATP required. And it leads to the formation of what? 2 molecules of NH3. Along with that, as I said, hydrogen definitely is there. Now what will happen? From ATP, 1,1-phosphate will be utilized. So 16 ATP will give rise to 16 ATP plus 16 PI, inorganic phosphate. PI stands for inorganic phosphate. So this is the reaction happening. Clear? So total of 8H plus and 8 for the electron transfer 2 ATP. 16 ATP after utilization becomes 16 ADP and 16 PI. This is very important. Now, coming to the next topic, that is nitrification. So after the ammonia formation, we see ammonia formed by the nitrogenase activity, right? Into 2 molecules of ammonia is formed. Now this ammonia will be converted into nitrite, and that nitrate will be absorbed by the plant. So for that, ammonia, it is converted into ammonium ion, and this ammonium ion by the oxygen is to bacteria. Name of the bacteria is Nitrosomonas and nitro. What will be formed? Nitrite, NO2. This is not a balanced equation, okay? Nitrite. Now this nitrite again in presence of nitrobacter will form what? Nitrate. This is nitrate and nitrite. And thus nitrate will be absorbed by whom? It will be absorbed by the plants. Now we will see how the nitrate is nitrate acylation. So in nitrate will happen, the nitrate will get converted into nitrite. In presence of a nitrate reduction. And called process nitrate reduction. Nitrate reduction. Second, gets converted into ammonia. Okay. what happens in presence of nitrite reductase enzyme. And process nitrite reduction. And from this ammonia, later what we say, amino acids. Amino acids are synthesized. So we will take the amino acids. Clear? So the ammonia, it is formed by the nitrogen fixation, by any kind of nitrogen fixation ammonia we are getting. That ammonia is that nitrate will be, and so later on nitrate assimilation will take place, whereby nitrate becomes what? Ammonia, and from ammonia we can get amino acid synthesis. We'll quickly do amino acid synthesis. Yeah, sorry for the gap in between. So next one, the amino acid synthesis. So there are two methods for the amino acid synthesis. Number one is reductive amination. Reductive amination. So in reductive amination, what is happening is the ammonia is converted into ammonia and that combines with alpha ketoglutaric acid, and we are getting glutamic acid as the product. And this ammonium ion, it combines with alpha KGA along with NADPH or NADH. In presence of glutamate, it gives rise to glutamic acid, which is an amino acid. Some kind of amino acids will be synthesized, which ultimately forms a protein at last. Our second one is transamination. Transamination. So transamination glutamic acid through a series of reaction can form some other amino acid. And it has not what here. Here the glutamic acid can also leads to the formation of some amides. It is having more nitrogen than any other amino acids. Okay, and usually these amides, they are translocated through okay. So that's all about this chapter. So one day the chapter started with the criteria of essentiality. We have seen the criteria, and after that we are having 17 essential elements. That 17 essential elements we classified based on various categories, whereby we detail the which is present, macro and micro, their deficiency symptoms we have seen, functions we have seen, toxicity also we have seen. Later on we have taken hydrogen effects, we have nitrogen assimilation. So hope this class you understood everything. So wishing you all the best for your upcoming NEET examinations.